All right, hello. Just a bit of background. COVID, as you all know, is a deadly virus that is wreaking havoc all over the globe. And while there are several pharmaceutical companies that have created vaccines against this virus, questions have been raised about their continued efficacy as new mutations of COVID arise. So, the aim of this study was to design an mRNA vaccine for a novel mutant of COVID-19, K417N. This basically just means that in the spike protein, there's an asparagine instead of a lysine at the 417 position. As you can see here in the middle of the page is the image of a coronavirus labeled COVID-19. Notably, this type of virus is coated with spikes. And these spike proteins are what the body recognizes and forms an immune system response against. In the bottom corner, you can see a close-up image of one of these wild-type COVID-19 spikes, which has been rendered in primal, and an image of the linear DNA domain map of the wild-type spike protein. That DNA from the wild-type spike protein will undergo 2 rounds of mutagenesis using primers with codons commonly found in humans. The first will be using this primer to induce the K417N mutation, and the second will induce these 2 proline mutations, which will lock the spike protein into its pre-fusion conformation and prevent random helical change. As you can see here in the linear DNA domain map of the mutant spike protein, both the K417N mutations and the 2 proline mutations are in red here, but the K417N mutation is in the RBD, which stands for receptor-binding domain. This is the domain that engages the host cell receptor and is important for ACE2 binding and antibody recognition. K417N mutation is a mutation which, when present in a vaccine, may allow the host to have a more complete and robust immune system response. Once these 2 rounds of mutagenesis are complete, the mutated linear DNA will be incorporated into a plasmid vector using the restriction endonuclease BAM-HI. In vitro transcription would then be used to test if there is a successful transcription of the mutated DNA into mRNA. Then HPLC will be used to purify the mRNA transcripts produced. In the bottom right corner, you can see the mRNA sequence that should be produced with AGXT UTRs. Future experimentation would include adding the mRNA transcript to a lipid capsule for nanoparticle insertion to see how well a mammal can produce and respond to the spike protein. Then research should be done to see how well this sequence could possibly work as a vaccine for humans against the K417N mutant. I'd like to acknowledge both my professor and their TA for all their wonderful help on this project. Thank you.